The present session deals with isolation of somaclones and its applications. In the previous session, I discussed about the causes of somaclonal variations. A range of somaclonal variations are useful in crop improvement programs. Example, drought tolerance, salt tolerance, certain disease-resistant lines in some crops, increase in number of tillers to increase the yield in rice plant, early flowering, etc. The recovery of these homoclones from the in vitro culture is of great importance and are then screened for their genetic stability in subsequent generations. The genetically stable somaclones are then multiplied to create new cultivars. Hence, screening and isolation of somaclones is a key step towards crop improvement. Isolation of somaclonal variants can be grouped into two categories: screening and cell selection. Screening can be done for detection of somaclones and is used in many cereal crops successfully. Here, a range of regenerated plants or cells, that is R1 or SC1, are used to identify variants, and then it is confirmed by use of R2 progenies. Example, screening for yield in a variant. Computer-based automated cell sorting devices are also used for screening, as many as 1000 to 2000 cells per second for desirable traits. Example, green and non-green pigmented cells can easily be selected through computer-based cell sorting. The variant cells are then screened and selected for regeneration to produce complete plantlets or somaclones. Selection of somaclones can be at two levels: the intact plant level and also in cultures. It could be in vivo or in vitro methods. The different selection strategies used for cell selection or somaclonal variants are positive, negative, visual, and analytical screening. Positive selection. In this selection method, selection pressure is applied on the cells, which lead to survival and growth of only the vibrant cells. If the selection pressure allows the growth of variant cells, it is known as positive selection. Example, in case of selecting antibiotic resistant lines, example for kanamycin, the media is supplemented with the compound kanamycin. Only those cells that are able to grow in kanamycin or resistant to kanamycin would be growing. Hence, these colonies or cells can be screened and selected. There are nearly 4 approaches of positive selection: direct selection, rescue selection, stepwise selection, and double selection. Positive direct selection method for selection of somaclones in vitro. This is the most common selection method used. Here, the somaclones are resistant to the selection pressure and hence they survive and divide. The wild type cells are susceptible and are killed by the selection agent. Example, the kanamycin resistant lines that I discussed in the earlier slide. This type of direct selection method has been used in isolation of cells which are resistant to certain herbicides, elevated salt concentrations, antibiotics, and amino acid analogs. Positive selection by means of rescue selection. In this method, the wild type cells are killed in the selection media. The variant cells or the somaclonals survive in the selection media but cannot grow or flourish due to the unfavorable environment. Here, after the selection is done, the selection agents are removed to recover these somaclones or variant cells, and it is then grown under normal media. This approach has been successful in recovering low temperature and aluminum resistant variant lines or somaclones. Positive stepwise selection. In this method, the selection agent is gradually increased in the media from a low to cytotoxic levels. The variants that survive in these stepwise selection are then isolated and checked for genetic stability. Example, to isolate salt-tolerant lines. Initially, low concentrations of salt are supplied to the media. The resistant clones are then selected and subjected to increased level of salt concentrations in subsequent steps. At each step, the resistant clones are isolated. Next, double selection method. In this selection method, two parameters are used for selection. One selection for survival and the other could be any other feature of these cells. Example, selection for resistance to antibiotic streptomycin. This also inhibits chlorophyll development in cultured cells. Here, the selection is based on cell survival in streptomycin media. First feature, as well as for development of green color colonies. The second feature. Negative selection to isolate somaclones. In this method, the selection pressure or selecting agent favors the growth of wild types. The wild types are then eliminated by counter selection pressure. Example, in a media supplemented with mutagens like chlorate and bromodioxyuridine, the dividing cells are eliminated. In this method of screening, the cells are first grown in a mutagen for a standardized period of time. This is then replaced by normal nutrient growing media. This leads to elimination of the dividing cells while survival of non-growing cells. This method is useful in selecting auxotropes. Next, visual selection. This type of selection is useful in isolating phenotypically visual traits. Example, cells that synthesize chiconin can easily be isolated and screened from non-developing chiconin cells. Analytical screening methods are utilized to screen and isolate somaclones for certain metabolites, bioactive components. In this particular screening method, the cells are first isolated and the component in question is isolated, extracted, and then checked and quantified by means of analytical methods and instruments like HPLC and GC. Somaclones can be isolated with or without selection pressure. Selection of somaclones without selection pressure. Here, the plants are regenerated by means of indirect organogenesis. The plantlets are then grown in pots and transferred to field and then screened for somaclonal variants. Here, in the nutrient media, there was no toxic or inhibitory substance or selective agent. Selection with selection pressure. In this type of selection, the cells are grown in a media containing appropriate selection pressure or selective agent. There is preferential survival or growth of variant cells in these specified selection media. The explants could be screened for certain toxins, amino acid analogues, environmental stress tolerance, etc. The mutants can be effectively selected. Steps to isolate somaclones by using selection pressure. In this method, the explants are first cultured in normal nutrient media to induce callus cultures. The callus are maintained by subsequent subcultures for increase in biomass. After these callus are then inoculated into media containing selection pressure, which could be antibiotics, herbicides, toxins, etc. The callus is then subcultured in these selective media, and after each subculturing, the tolerant lines are selected. The isolated tolerant lines are then finally induced for plant regeneration. The regenerated plants are then screened for the somaclones. The figure on the right shows steps to achieve disease-resistant plants against a particular pathogen. In this, the selection media consists of toxins isolated from a particular pathogen. The calli are screened for these toxins. The tolerant lines are then made to induce plant regeneration. The regenerated plants are then screened for genetic stability. In this way, agronomically important disease-resistant trait in plant is produced. Examples of certain agronomically important somaclones achieved by means of selection pressure. Applications of somaclonal variations. Somaclones are a useful source of introducing genetic variations in plants. Some of these could be for agronomically important traits like resistance against certain pathogens or environmental stress. Some of the examples I have mentioned in my earlier slides. In certain cases, somaclones are being standardized and developed for increased secondary metabolite production. Example in case of chiconin synthesis. Disadvantages of having somaclonal variations in tissue culture. Somaclonal variation is an undesirable trait in case of micropropagation of elite genotypes or genetic transformation. In micropropagation, the idea is to get clones. Somaclonal variants sometimes show undesirable features such as reduced fertility and growth rate. Certain somaclonal variations are genetically unstable. These are unpredictable also. In certain cases, somaclonal variations lead to loss of certain important agronomical traits. Somaclones may not occur in certain complex agronomically important traits like yield. Some of the somaclones may have pleiotropic effect. Somaclones require extensive field testing before they are introduced as new lines in crop improvement programs. Generally, sexually reproducible characters are preferred. Thank you.